Hi everyone, in this video I'm going to give more background behind the latest publication from our lab. And in this work, where we were collaborating with MIP Diagnostics, we were using nanoparticles produced for them with high affinity for COVID-19 using our thermal detection method in order to facilitate fast, robust and reliable detection of COVID-19. And you can find a link to the paper here via the assays sensors website. Now, the first thing that you might ask yourself is why would we want to replace COVID-19 tests? So we have lateral flow tests at home or rapid antigen testing, and this works relatively fast. It's very easy to do. Everyone can do it themselves. But the problem with that is that even though it's very good at picking up when someone has a high viral load, so when you're positive, it does miss a lot of the earlier infections. And that's because the sensitivity is not quite good enough. Now, there's other reasons for why we want to replace such a test. So if you think in general of tests where we use antibodies, and the problem with these antibodies is that they are relatively expensive, but they're also not quite as robust as we would want. So you would need to use them in combination with a specific buffer. We would need to use them at a specific temperature as well. So it means they have a limited shelf life, and they might not work as well in hotter climates, for instance. Besides that, there's also still a large portion of antibodies that, even though we can produce it with recombinant antibody technology, that are animal derived. So it means that we use animals such as mice and rabbits in order to produce them. And actually this is such a problem that in the EU alone we still use approximately 1 million animals per year just for this diagnostic testing. So the reason for moving from this test is partly because of ethical reasons, so to promote animal welfare, but also that it might enable us to measure COVID-19 in scenarios where normal antibody-based tests can't work. So for instance, at elevated temperatures or in other challenging diagnostic environments. I've done some previous videos on molecular imprinted polymers, so if you're interested in that aspect, I recommend that you go to this video in the link above. But to just generally recap, molecular imprinted polymers, or MIPs in short, are materials that are able to rival the affinity of antibodies, but much cheaper. They're versatile, so they can be used for small ions going to pharmaceuticals, to larger macromolecules. And very importantly, they're produced completely in an animal-free process, and they're very robust. So it means they can withstand extremes of pH and temperature, which we'll show later on. And here you can briefly see how this works. So we cover our probe or our electrode with this MIP material, and this is done by having a templated polymerization, and after you remove the template you end up with binding sites that resemble the size and the shape of what you want to detect, but there's also a chemical interaction between them. So essentially you get a highly porous material with high affinity binding sites. So even though molecular imprinting has been around for quite a while, it wasn't until the 90s that this really taken off, because then they started looking at non-covalent imprinting. And at the beginning of my career, I actually used a process where we produce microparticles, which generally lack in affinity, are not quite as good as the nanoparticles that we work with at the moment. But what you can briefly see here in this animation, what you would get is that you would have a microparticle with these binding sites, and as soon as you get binding to it, you would see a change in electrical resistance. When I started my independent research career, a big drawback of these microparticles is, even though it's quite easy to produce them, is that for a lot of clinical measurements it's not quite as suitable, because they can't reach the limits of detection that you want, especially in medical research. So here is where we partnered up with MIP Diagnostics, and there we started using a very innovative solid phase approach to produce these materials. And in brief, you can have a look at the papers below if you want to know more about it. Here, rather than eluting off your target, you begin by attaching your template to a solid phase, you polymerize around it, and then you elute off the polymer material instead. And the big advantage of that is that you can use your solid phase as an affinity medium. So you end up with very homogeneous particles, and only the ones that have a very high affinity, which is what you will get when you do this temperature elution. Now in this project, we obviously applied this approach to COVID-19. And what they did is use a specific epitope, so they didn't work with the virus in order to produce a material, which also inherently makes it a lot safer. but they use a specific epitope within the receptor binding domain. And that actually has advantages, even though we know there are lots of mutations that might occur in the receptor binding domain of the spike protein in general, because we only work with a very small fragment of it that actually work to our advantage, because it's less likely that you'll have mutations within the short fragment, which is around 10 to 12 amino acids that was selected. The other opposite advantage is then when you use with like an epitope, it's much more stable and it's a lot lower in cost, so you can actually work with quite big quantities. What we did then is we've taken these nanoparticles and functionalized them into screen printed electrodes. And screen printed electrodes are very often used commercially, they're very low cost and they're disposable electrodes, so they're very suitable for commercial research. And as you can see here that we used our specific setup, which is based on this thermal detection, so of the heat flow that goes between the surface. As soon as we see that there was binding to these nanoparticles, there was a change in the thermal resistance. And we are working here with 3D printed measurement cells, and the initial cells that we use still required like a volume of 2 to 3 milliliters, which is quite high if you want to actually work with clinical samples. But within this project, we successfully managed to redesign them in order to work with samples as small as what you get from a normal swab from your nose. Now, one of the key advantages, which I mentioned before with these nanoparticles, is not so much that, yes, we will be interested in the affinity and how good they are at binding the virus, but the robustness is very important. So what they looked here is looking at different conditions where antibodies would struggle with. So first of all, you're able to autoclave the material. So these nanoparticles, they come in a water-based solution. So when you autoclave it, you basically essentially sterilize it. So you steam it, so it goes up to really high temperatures. So you could determine the affinity before and after, and could see that nothing was really changed. Obviously this is very beneficial because it means that you might be able to store the material for a very long time, even when it's in solution. Then also using atomic force microscopy, we showed that there really wasn't a change in the shape of the nanoparticles when we exposed it to elevated temperatures up to 50 degrees or to extremes of pH. Because we knew there were problems of when you, for instance, expose antibodies to acidic media, such as what you've seen in lateral flow tests when people were using, for instance, Diet Coke to interfere with the measurement results. And clearly it turned out it wasn't just the shape that wasn't affected, but it also turned out that these nanoparticles remained the affinity even within these conditions. And that eventually might open up some interesting prospects if we wanted to measure in some challenging environments. So we had to redesign it to an addition type of setup where we actually only added the clinical samples with a pipette. And what we then figured out, and this is based on some modeling results that we looked at, is because you're not flowing through the system, you actually have less movement in the system. So by adapting it to having like a system that's not moving, you also create less noise in the system. So it was like a twofold. It was much better in terms of the clinical samples because we're able to measure really small volumes up to like 100 microliter. And also it reduced the noise because like the liquid was completely still. So if it was moving somewhat, you would see some interference in the thermocouples that we use for the thermal readings. And the advantage of using these prototype devices, sometimes we've made some devices in Perspex which are longer lasting, but particularly in with 3D printing, you're able to develop disposable systems. And when you work with biomaterial or material that might contaminate something, it's very beneficial that you can throw away both the electrodes, because remember these were these really disposable, cheap screen printed electrodes, but also dispose of the 3D printed setup and just use a fresh one every single time. Now let's have a look at the measurement results. So what we essentially see is that when you have binding within the material, you start blocking the cavities that you've got on the surface. And when you block it with something which has a lower conductivity, in essence we see both an increase in the electrical resistance, but also in the thermal resistance. And this thermal resistance is what we relate to as RTH. So in essence, if you've got a reference material, then you would see you will add something to the solution. You see this little spike because you add in a cold liquid, but in essence, nothing really changes of the signal. Whereas if we are working with COVID-19 or samples that are spiked with COVID-19, we would see a gradual increase. So gradually you would see the signal going up. And we can use this to construct a calibration curve and actually determine how much it would go up if we have a known amount of antigen. Obviously, you need appropriate references, so you can look at, for instance, the non-imprinted polymer material, which we did, but we also compared our results to antibodies, and you could see that the results were definitely comparable or probably somewhat marginally better. And then we also looked at solutions where it wasn't just COVID-19, but we also looked at other common interferents that are present, such as, for instance, you have high glucose levels in your blood, but also some other markers that might indicate inflammation. And as you can see here, that our sensor was only really responding when there was COVID-19 in the sample. Now, all these tests were initially done in buffered solutions, which are relatively easy to measure before we move to the clinical work. And if you want to compare a sensor, and here, like on the right diagram, you can see how it compares to other literature. So in the green bit, we've got our own sensor. The blue is the commercial lateral flow tests, so the one you probably have at home, and the red is what else has been reported into literature. So you can see that the sensitivity, what we have, is much better than a commercial test, and it's definitely comparable to other reports that are already out there in literature. But you have to imagine that big advantages of our test is that it's really fast. We can do this in around 15 minutes, very low cost materials, and also this fully disposable setup, which is very beneficial. Now, besides buffered solution, the next step is obviously be to look at clinical samples, and this is really important because especially when you've got this swab material, which is somewhat viscous, and then it goes into a specific medium, and then you also get heat treatment to it, so it's safe to work with, so you can use it under class 2 requirements. This has a lot of complications, and it comes with like some matrix that might lead to a lot of non-specific binding to your surface and therefore can interfere with your signal. Now, what we did is we looked at 10 clinical samples. Half of them were of positive patients with a known viral load, as was determined with PCR, and then the other ones were 5 negative samples. Now, as you can see in general, so there was like a range of what the viral load was in the samples, but generally you can see a very clear trend between negative results and positive results. What are the next steps for this? Well, here you only see 10 clinical samples, which is obviously not enough to really validate our technology or to establish a correlation with what's already out there. So what we need to do is obviously measure more samples, but also more interestingly, we want to look at new and emerging variants because it's obviously constantly changing as we go along. So what you've seen in the previous slides were results from the Delta variant, but now we're quite keen to also look at the Omicron variant, and who knows what other variants might come up. So we'll be investigating this, but also we would like to make some changes to the setup. So what we use at the moment is that we've got a device which is coupled to a laptop. So we still need to impose like a temperature gradient and run like a computer program to use it. But within our lab, we also have materials that are very sensitive to changes in temperature. So these materials, they actually change electrical resistance. So rather than measuring temperature changes, we're interested in just looking at the electrical resistance itself. So that would go towards like handheld device, where it would look something like, as you would see in the slide, you would simply dip it into a solution and you would get a very quick reading. Now, research you never do alone, so there was a large team working on this, but the majority of the experiments was done by Jake McClements. Now, we also worked with researchers at MIP Diagnostics that I mentioned before, in particular Francesco Conferotta, and we also work with long-standing collaborators at Manchester Metropolitan University in the UK and Université Libre de Bruxelles in Belgium. Now, if you would like to know more about the work or have a look at, there were also quite a couple of press releases done on our work, and have a look at the description in the video to find out more. Thanks for watching.